Have you tried having colds, food poisoning, chickenpox, or taking antibiotics? Now, these are caused only through microorganisms. So they are found in skin, deep sea hydrovents, the stomach, our mouths, gadgets, the soil, and even in the air. Most are harmless and they protect the skin, airway, and digestive system from pathogens. Pathogens are microbes that cause diseases. We are going to be tackling about the humans and the microbial world. This will be composed of 3 parts. We are going to tackle the dispute over spontaneous generation. It all started with a father and son, Hans and Zacharias Janssen, in the late 16th century. The microscope was composed of a tube with lens at its top and bottom, with magnification of 3 times to 9 times, and it's quite blurry. A cloth merchant turned biologist, Anton van Leeuwenhoek, made the discovery of microorganisms. He first made an instrument to see into his clothes, since he was a cloth merchant, and accidentally discovered bacteria, millions of animalcules in a single drop of lake water as well. This also led to the germ theory by Friedrich Henle. Where did the microscopic forms originate? Now, they initially thought it was spontaneous generation. So what is spontaneous generation? It is where life emerged from non-living materials, such as emergence of worms from this decaying meat. Theorists and scientists have been debating over it for years, leading to the dispute over spontaneous generation. This will be composed of early experiments, the experiments of Pasteur, experiments of Tyndall, the golden age of microbiology. This is the summary of the dispute over spontaneous generation, challenged by Francesco Redi in the 17th century. Francesco Redi disproved this idea, and according to his experiments, worms actually come from the eggs of flies and not from the meat itself. Still, people didn't believe in him, for it wasn't convincing enough to be an evidence that microorganisms didn't arise from spontaneous generation. Next, John Needham. He used flasks containing broths sealed with cork, and microorganisms emerged. He said something like, yes, they really do arise from spontaneous generation. But then, Lazzaro Spallanzani did quite the same experiment, but the difference is he boiled the broth longer and sealed the flasks by melting the glass necks closed. So he did not agree with John Needham. He made almost the same experiment, but longer duration of boiling the broth and close the flask by melting the glass. So no bacteria appeared. He said that microorganisms from Needham's experiment entered the air, and the corks wasn't enough to keep them out. Louis Pasteur. He also disproved spontaneous generation. He proved that the air does contain microorganisms. He trapped them in a cotton plug and dropped into the sterilized broth. It came cloudy, and a sign of microorganism growth. He also became the father of microbiology. The experiments of John Tyndall. In his experiment, some microbial forms are not killed by boiling. With Ferdinand Kohn, he discovered endospores. These are the heat-resistant forms of some bacteria. Shown here is the different stages of endospores, or the heat-resistant bacteria. The dispute over spontaneous generation, it led to the golden age of microbiology. The discoveries led to the acceptance of the germ theory of disease. Diseases come or originate from the emergence of germs. So there is the scientific method. First is observing an occurrence and asking a question about that situation. Second, developing a hypothesis that explains the occurrence and devising an experiment that tests the hypothesis. Third, doing the experiment, collecting the data, and drawing conclusions. And lastly, communicating the results, methods, and conclusions. What is a scientific theory? It is an explanation supported by a vast body of experimental evidence. Microbiology, human perspective. Under these would be the vital activities of microorganisms, the commercial benefits, as research tools, the microbes, and also the diseases across generations. The vital activities of microorganisms. Microorganisms are masters of recycling, like recycling nutrients, replenishing oxygen, converting nitrogen gas into usable forms, and also degrading materials that others cannot. For example, cellulose in plants cannot be degraded easily. Microbes does the work for you, especially microbes in the stomach of animals and humans. The commercial benefits of microorganisms are food production, biodegradation, Commercially valuable products, biotech, and research tools. Commercial benefits of microorganisms, as you can see, pharmaceuticals, baking, dairy, beverages, poultry, poultry feed, biopolymer, paper and pulp, leather, textile, cosmetics, detergents, organic synthesis, and waste management as well. Food production, yeast is used for bread and for fermentation. Milk and probiotics, these are used for yogurt, cheese, and buttermilk. Biodegradation, now this is also known as bioremediation. So biodegradation of products have a big potential in reducing our waste. We also do have our commercially valuable products, the cellulose. These are used in headphones. The hydroxybutyric acid used in manufacture of disposable diapers and plastics. Ethanol used as biofuel. Hydrogen gas used as possible biofuel as well. Oils used as possible biofuel. Insect toxins used in insecticides. Antibiotics used in treatment of diseases. Amino acids used as dietary supplements. Now, these can actually be made synthetically. But then, in microorganisms, this is time efficient and also cheap. That's biotechnology. It is where genetically engineered plants are produced, micro-produced insulin as well. Now for genetically engineered plants, now see the difference of the modified and unmodified plant. For the first is the unmodified plant. Let's look at it. So it uses less energy for photosynthesis and more energy lost as heat. In modified, 15% in photosynthetic efficiency, therefore 15% more plant growth. And also it uses less energy and it is lost as heat. For micro-produced insulin, so this is an example. How do they make insulin from recombinant DNA? So first we have the fermentation tank, and then the recombinant bacterium produce insulin, and there the insulin is harvested. Microbes as research tool. Now microbes have been a tool for major advances in understanding life in study of microbes. How did microbes improve our research about life in Earth? First, microorganisms have fundamental metabolic and genetic properties as higher life forms. Second, experimental results are obtained instantly or quickly. Microbes and diseases. Trend in death rates due to infectious diseases throughout the years. Now, here, this is an example of crude death rate for infectious diseases in US per 100,000 population per year. The pathogens cause disease, but the death rate from infectious diseases has declined over the past 100 years. 40 states have health departments. Now this, the highest part, that would be the influenza pandemic. Now this is the last human-to-human transmission of plague. And then the first use of penicillin, the first polio vaccine introduced, and the passage of Vaccination Assistance Act. Due to the discovery of antibiotics and vaccines, the death rates of infectious diseases have dramatically decreased. Here we see the past triumphs, the remaining challenges, and other chronic diseases. The past triumphs, we have discovered antibiotics vaccines for pathogens like the smallpox, the plague, polio, and epidemics. And also, epidemics can be found in animals and plants as well, not just in humans. For plague, we have used antibiotics. Smallpox virus, we have used vaccines and antibiotics. For polio, we have used vaccines. Then the remaining challenges would be EID, also known as the emerging infectious diseases, the MERS, Hep C, SARS, influenza, emerging Lyme diseases, AIDS, hantavirus, E. coli, and tuberculosis. Do you know that these infectious diseases can spread again? So what are the factors making infectious diseases common? First, people no longer have knowledge of the dangers of the diseases since it happened a long time ago. Second, misinformation about the diseases. Irrational fears of vaccines. And then the pathogens become resistant to antimicrobial medicine. The pathogens become resistant to antimicrobial meds, for example, the tuberculosis and malaria. Others are changes in characteristics like growing old, having AIDS and tuberculosis, and travelers carry pathogens around the globe. The chronic diseases, stomach ulcers, and the chronic indigestion. Now, they thought that it came from stress, but then it actually came from bacterium. So the treatment would be antibiotics. For cervical cancer, it has been related to HPV infection. That's why it can be prevented through the HPV vaccination. Microbes and diseases. So to prevent the spread of diseases, there are vaccinations and antibiotics. Here shown in the picture is the new and newly recognized infectious diseases or disease agents in humans and animals since 1976. So as we can see, here's the E. coli in United States, AIDS, the Legionnaires disease, swine flu, cryptosporidiosis, the hepatitis D, C, hemorrhagic fever, Ebola virus, Vibrio cholerae, Malaysian encephalitis, human and equine morbillivirus, avian flu, human T-cell virus, hand-time virus, severe acute respiratory syndrome or the SARS in China, and then the Saudi Arabia, the MERS. What are we supposed to do to prevent the spread of diseases? That would be the use of vaccination and antibiotics. We also need to prevent emerging infectious diseases, and some chronic diseases are caused by microorganisms. That concludes the microbiology in human perspective. Up next, we are going to learn about the members of the microbial world like viruses, bacteria, fungi, and parasites.